Have you ever walked into a room and smelled freshly baked cookies? Or maybe you've smelled the sweet scent of a flower? Your nose is an amazing chemical detector. Well, in the world of science, we have a machine that acts like a super-powered nose. It's called a gas chromatograph, and it's one of the coolest tools we have for figuring out what's inside a mixture. It can separate, identify, and measure all the different ingredients, even when they are jumbled together. It's like being able to look at a cookie and instantly know the exact recipe, down to the last sprinkle. So what is gas chromatography? Let's break it down. Chroma means color, and graphy means writing. The name comes from an early experiment where scientists separated plant pigments, creating colorful bands. Gas tells us that this technique works with substances that can be turned into a gas or vapor without being destroyed. Essentially, gas chromatography is a powerful scientific method used to separate the different components of a sample. Imagine you have a smoothie with strawberries, bananas, spinach. Gas chromatography is like a magic strainer that can pull each of those ingredients out, one by one. Why is this so important? To understand how this science magic works, we first need to meet the machine itself, the gas chromatograph, or GC for short. A GC might look like a complicated box with buttons and tubes, but it's really just made of a few key parts working together as a team. The first part is the gas supply. This is usually a big tank of a very pure, non-reactive gas, like helium or nitrogen. We call this the carrier gas because its job is to carry our sample through the machine. Think of it as a river, and our sample is going to be a little boat floating down it. The carrier gas needs to be steady and pure so it doesn't interfere with our results. Next up is the sample injector, or the inlet. This is the starting gate for our chemical race. The sample, which is usually a tiny drop of liquid, is injected here with a syringe. The injector is super hot, typically hundreds of degrees Celsius. This heat instantly vaporizes the liquid sample, turning it into a gas. It's crucial that the entire sample turns into a gas at once so that it can be swept into the system by the carrier gas. The injector is designed to introduce this puff of sample gas in a very quick, clean, and repeatable way. It's like dropping a perfectly formed paper boat into our river at the exact same spot every time. The heart and soul of the gas chromatograph is the column. This is a very long, very thin tube, often coiled up like a garden hose to fit inside a temperature-controlled oven. The inside of this tube is coated with a special substance called the stationary phase. As our gasified sample travels through the column, its different components will interact with this coating in different ways. Some will stick to it more, and some will barely stick at all. The oven that holds the column allows us to precisely control the temperature, which is a key factor in how well the separation works. Finally, at the end of the column's long journey, we have the detector and the data system. The entire process begins with a tiny, carefully measured amount of our sample. Let's say we're testing a drop of vanilla extract to see what flavor compounds are inside. A scientist uses a special tool called a microsyringe to suck up a very small volume of the liquid, often just a microliter, that's 1/1,000,000th of a liter. The syringe needle is then pushed through a rubber seal called a septum on the sample injector port. With a quick push of the plunger, the vanilla extract is injected into the GC. This step needs to be done quickly and smoothly to make sure the entire sample enters the system as a tight, compact band. Once inside the injector, our drop of vanilla extract meets a blast of heat. The injector is kept at a very high temperature, hot enough to instantly flash vaporize the liquid into a cloud of gas. Vanillin, guaiacol, and others are now floating around as individual gas molecules. At this exact moment, the carrier gas comes into play. A steady stream of an inert gas like helium is constantly flowing through the injector. This stream of helium acts like a gentle but persistent wind, immediately picking up our cloud of sample gas and sweeping it away from the injector and toward the next part of the machine, the column. The choice of carrier gas is very important. We use inert gases, which means they are chemically lazy and won't react with our sample. Helium is a popular choice because it's light and allows molecules to move through it quickly, leading to sharper results. Nitrogen is another option because it's inexpensive, but it can sometimes be slower. The key is that the carrier gas must be pure and flow at a constant, controlled rate. Think of it like a conveyor belt moving at a perfectly steady speed. If the speed changed, it would mess up the timing of our separation. Now for the main event, the journey through the column. This is where the actual separation happens. The mixture of our vaporized sample and the carrier gas flows into one end of the long coiled tube. As we mentioned, the inside of this tube is coated with a stationary phase. This can be a waxy liquid or a solid material that is bonded to the inner wall of the column. The different chemical compounds in our sample will interact with this stationary phase in unique ways. It's like running an obstacle course where some runners are better at certain obstacles than others. This interaction is the secret to separation. Imagine our vanilla extract sample contains two main compounds, a sticky one and a non-sticky one. As the carrier gas pushes them through the column, the non-sticky compound doesn't interact much with the stationary phase. It spends most of its time in the moving carrier gas, zipping along quickly. In contrast, the sticky compound is more attracted to the stationary phase. It will repeatedly dissolve into the coating, hang out for a moment, and then evaporate back into the carrier gas to move a little further down. This stop-and-go process slows it down significantly. The temperature of the column, which is controlled by the oven, plays a huge role in this race. Increasing the temperature gives all the molecules more energy. Scientists often use a temperature program, where they start the oven at a low temperature and gradually increase it over time. Because of these different interactions, our initial puff of mixed gas begins to separate into individual bands as it travels down the column. The length of the column, often 30 meters or more, gives them plenty of time and space to separate properly. After the long and winding journey through the column, the separated components, still carried by the stream of helium gas, finally exit and enter the detector. The detector is the finish line, and its job is to announce the arrival of each chemical as it emerges. It can't see the molecules like we do, but it can sense their presence in other ways. There are many different types of detectors, each designed to be sensitive to different kinds of chemicals. One of the most common types of detectors is the flame ionization detector, or FID. The FID has a tiny hydrogen-air flame burning inside it. When a carbon-containing compound exits the column and enters the detector, it gets burned in the flame. This burning process creates charged particles, or ions, which allow a small electrical current to flow between electrodes. The detector measures this tiny electrical current. The size of the current is directly proportional to the amount of the substance that is being burned at that moment. So when a big band of a separated compound flows through the detector, it generates a large electrical signal. When nothing but pure carrier gas is flowing through between the bands, the signal drops back down to zero or baseline. The detector is incredibly sensitive and can detect minuscule amounts of a substance, sometimes down to the picogram level. That is a trillionth of a gram. This electrical signal is the raw data from our experiment. The detector doesn't know what the chemical is, but it knows when it arrived and how much of it there was. This stream of electrical information, a series of peaks over time, is then sent to the data system, which records and plots a graph called a chromatogram. X-axis equals time, minutes, the retention time, y-axis equals detector signal strength, which corresponds to amount present. Each component passing the detector makes a peak. Peak position identifies the compound. Peak size tells how much is present. For example, a vanilla extract chromatogram shows several peaks, vanillin and other aroma molecules. A peak at 5.3 minutes is compared to a compound library. If pure vanillin matches 5.3 minutes, that peak is vanillin. Calculating peak areas gives exact quantities or percentages. The chromatogram is a chemical fingerprint. So this super sniffer technology is really cool, but where do we actually use it? The answer is almost everywhere. One of the most important applications is in environmental testing. Scientists use gas chromatography to keep our planet safe and clean. The GC can detect tiny trace amounts of harmful pesticides, industrial chemicals, or oil spills. In food and beverage, it ensures quality and authenticity, distinguishing natural vanilla from artificial flavors. It's used to analyze coffee aromas or measure alcohol content in beer and wine. In forensics and medicine, GC helps identify suspicious substances, fibers, paint chips, or traces of accelerants. Paired with a mass spectrometer, GCMS is the gold standard for drug testing in sports and clinical labs. In petroleum, GCs analyze crude oil and separate it into gasoline, jet fuel, and valuable fractions. Even space missions used gas chromatographs. The Viking landers carried one to analyze Martian soil. While a real GC is sophisticated, you can see the principle with a simple paper chromatography experiment. Paper acts as the stationary phase. A moving liquid is the mobile phase. Here's what you'll need: a coffee filter, some non-permanent markers, a tall glass, water or rubbing alcohol, and a pencil. Cut a strip. Draw a dark line about 2 inches from the bottom, pour a little liquid in the glass, and suspend the strip so the line stays above the liquid. Watch as the colors separate. Black ink separates into blue, red, yellow, the same separation idea as GC, but visible and colorful. This is chromatography in action, a colorful, hands-on introduction to the concept. So, there you have it. From the starting line at the injector to the finish line at the detector, gas chromatography is like an incredibly precise and powerful chemical race. It helps answer questions about water safety, air quality, drug testing, and the purity of medicines and food. A guardian of quality and a protector of our health. From a simple paper strip to a high-tech GC on Mars, the core idea is the same: separate by sticking and moving. It's a testament to human ingenuity and our endless curiosity to understand the world around us. The gas chromatograph is one of science's unsung heroes, a super sniffer that reveals secrets one molecule at a time. And that, in my opinion, is just plain awesome. Science rules!